So this is the first part of the Aquaexcel repository training, and it will be organized in this way. So today we will try to show you something about the system which was developed within the first Aquaexcel project, and try to show you how to prepare very simple protocol and how to use that. In the second day, which is on Wednesday, we will try to go through the second part, where we will show you how you can describe your particular experiment, how you can attach your experimental data to the protocol, and how to manage your own protocols. In the last day, which would be on Friday, we will try to show you the extended features of the system. It means the plugins and modules for data processing and for communication with the measurement devices. And that should be the final part. We also expect that you will have some homeworks. That's why we have 1 day in between each part, where you can try to play with the system. You can use your own data, your own protocol, and you will see if there will be any problems during when you play with the system. So the things related to the organization, hopefully you can see how to use the Join Me conference system. So probably all of you can send a message to us. And the session is recorded, so it means that after the 3 training courses, the video from the sessions will be available on Aquaexcel webpage. So then you can play the video and you can follow it. If you will not catch everything during the training course, then you can go back to the video and try to reproduce all the steps. So the way how we will try to show you how to work with the system will be that on my screen I will show you every single step, where to click or where to write things, to show you on particular example how you can use the system for your work. The main idea of the training course is that the people who are involved in Aquaexcel project or in one of the transnational projects can use the system to describe the experiments or describe data processing, store the data on Aquaexcel server, and use all the features of the system to support the experimental work. So during the 3 days, you should learn what is the system about, how you can use it for your own work, and what are the benefits of the system if you decide to use it. The system is for free for all the people involved in the Aquaexcel project, also for the people who apply for a TNA project, Transnational Access Project, and then you can freely use the system for your own data. So before we will start, in the email, in the information about the training course, it was mentioned that you should prepare your own experimental protocol and your own data set. I will explain you why, because we will try to create, or you will try to create your own protocol. And the best way is to work with some real examples. So that's why it would be great if you can prepare some very simple protocol, some data set, and then we will try to upload everything to the system. From the point of view of the data security, you will see that each user has its own account, which is secured by the login name and password. So you need to be scared that someone else will use your data. But of course, it's better to use some data set which can be shared to the others. um, so this today we will start with the presentation about the system. Well, I will try to show you why the system was developed and what are the main features of the system. It should be the first 20 minutes. And then I will show you how to work with the software that you hopefully installed on your computer, and with the web interface, which is the second access point to the Aquaexcel repository. At the end of the course today, I will show you how to create your own protocol template. And at this point, we will finish because you will know how to create the protocol in the software. And tomorrow you can try to create your own protocol template. And on Wednesday, we will use this template to fill it by description of experiment and to attach experimental data to the protocol. Okay, so right now, I will use the presentation about the system to show you the basic functionality of the system and how the system is designed and what was the reason that the system exists. So the system is called Aquaexcel repository, and it's based on the solution called BioWES, which is the scientific data management solution. Which should serve for the management of experimental data and also the protocols, because we believe that only the experimental data together with full description of the experiment is useful from the reproducibility point of view. Okay, here's just a summary what I already told you. So in the first day, we will try to go through Aquaexcel repository, introduce you how the system works, and then you will have the homework to create your own protocol template. And the second day, you will fill the protocol, we will attach experimental data, and we will try to manage our protocols. And on the third day, we will try to explain you what are the special features of the system, the modules, the plugins, and how you can use the system through your mobile phone. And also, I will try to mention some standardizations that were created during the first Aquaexcel project and show you how it can be used in the system. So, what is the Aquaexcel repository? Aquaexcel repository is simply the server which is sitting in our institute. And we have some software tools like the software you installed on your computer and web interface to access this repository. How it can be used? It can be used for all the transnational projects, which means that once you apply for the transnational access project, then your account will be created in the system. And your application form will be moved to the system, so you can simply see everything in the system. I will show you how it looks like later. And you can simply start from this point to upload the data from the project, describe the protocols and the experimental work. Also, the results from the work packages of the individual parts of the Aquaexcel 2020 project can be stored in the system. And the system will be used for collaboration between the people who are involved in the Aquaexcel project. So the reason why it was developed was that there was a need to cooperate in between the people participating in the first Aquaexcel project, which already finished last year. And mainly the concept was developed to support the transnational access projects. which are the projects where every people, it's necessary that the people are involved in Aquaexcel projects can apply for this type of infrastructure project, can freely use the infrastructures of the Aquaexcel participants. So these are the transnational access projects. The second point why we developed the system is the reproducibility issue. So probably all of you know that the reproducibility is the high topic these days, and we try to develop the system to support the reproducibility. It means that we try to support the users to describe the experimental work well and link the description of the experiments together with experimental data. So it means that we have the complete information about what we did during the experiment. In 2015, there was the study published where they tried to identify the issues and the reproducibility, and one of them was the missing infrastructure for experiment management. And this is exactly the point where we try to help because the system is focused on the management of the experiments. As you can see, there were different issues with the standardization, the terminology, the procedures, and so on. So we tried to support all these things in the system. And during the training course, you will see how you can use the standardizations and communicate with the measurement devices. On this slide, I tried to just summarize what the data management process means. So first of all, definitely we have to measure something. That's what we know very well. All of us probably did some experiments, so you know that we have to measure something, and then we should describe the experiment. It means that once someone will try to reproduce the experiment you did, he should get very similar results. And here is the description of experiments is very critical because you should describe all the conditions that can influence the result of the experiment. Definitely, we would like to store the data at least, and of course the description of the experiment in the protocol. Probably we would like to use the standardization because usually the people cooperate, and once we don't use the standardized terminology, then it can lead to misinterpretation. Then we probably would like to organize our experiment so don't lose the track of the records. And we would like to link the protocol and or the processing steps in between each other. Definitely, some part of the work is to share our work. It means the data and the description of the protocol with the people to whom I would like to cooperate. And it's very useful to do the search because after 1 year, if we try to search what we did, it can be very complicated if we have just the paper version of our protocols and the data are stored somewhere on the hard drives, and we don't have the full control of that. Because I mentioned the standardization, then I just prepared one slide about the standardization to let you know that there are different initiatives on internet, for example, BioPortal or UL Ontology. And these initiatives try to summarize the terminology and standardizations from different fields of the research. So if you are interested in if some standardization or terminology exists in your field of view, then you can simply go to these web links and try to check if something exists. Our system can support the terminology from these portals, And on Friday, we will show you how it can be done. In 2012, we started the Aquaexcel project with a survey where we tried to learn what is the practice in the management of the experimental data and experimental protocols. 18 partners were involved in the survey around Europe, and we found three main outcomes of the survey. And we have learned that the experimental protocol design, which is related to the experimental world, is very good, as we expected. The standardization, usually we saw that the people are using the local laboratory standards. It means that it was some mixture of the standardized terminology and the terminology of the laboratory. So usually the people didn't know that there are some EU initiatives which try to collect the standardization from different fields. And this is one of the weak points of the data management. Data storage, it was very poor because the most sophisticated solution was the centralized storage on the server, where each person has its own directory, and this person can store the data there. Usually, the people are using the personal computers or personal hard drives to store the data from the experiments. And then it can be very messy because after some time the people lost the track where are the data from the particular experiment. Data sharing, the people usually use the emails or FTP servers to share the data. So therefore we try to support the sharing possibility in our system. And in general, the main outcome was that there is a lack of tools to support the data management, and therefore we try to develop the system. We will try to show you how it works today. Because our solution is not just the one solution you can use for data management, I try to prepare here the list of the possible solutions you can use. Probably you know the Elixir initiative or maybe Seek. These two projects are mainly focused on data storage, data processing, and the support of your work start when the data are measured, collected, and you can upload your data to Elixir servers and try to ask for the Elixir services to process the data, use data management tools, and so on. The systems called LIMS systems, which stands for Laboratory Information Management System, are often focused on the management of laboratory resources, and they are usually highly focused on some specific experiment, and they just describe this experiment. So this is the disadvantage of the system. Also, there are some tools called e-notebooks, which can very well describe the experiments. The problem is that e-notebooks usually describe the experiment in free form, which is not the best way for later automatic data processing because the system needn't to understand the description of the experiment. So therefore, we developed the BioWES system, which starts with the support from the design of the experiment protocol till the sharing. So it should help you in with all the aspects of the data management. So here's the just the point what the BioWES system and of course Aquaexcel repository is. So it's the scientific data and metadata management platform. This is important because we don't deal just with the data, but also with the metadata, which we understand the description of the experiment. The focus is on the reproducibility of the experiment, and we try to support the people from design of protocol to the sharing. We have two main parts of the system. The local part, which is usually the central server, where the people can store all the data. And then the central part, where the people can share the description of the experiments with the others. And it serves as the announcement of what the people did. So no experimental data are stored on the central system. So on this slide, you can see how is the system logically divided into three parts. The core part of the system is the local storage, which you can imagine as the server, which is for the Aquaexcel repository server. It's sitting in Czech Republic at the BioWES Institute. And what you can do with the local storage is that every single user has the account on the server, and you can do the management of the protocols on the local storage server. So you can do the queries, you can do sharing of your data and description of experiments. You can define the access levels of to your data. You can also do some visualizations, so you can see the evolution of your protocols, and you can do the data processing on the server. The second part, as I already mentioned, is the central storage, which you can imagine as some server again. But the difference between the central storage and the local storage is that on central storage there are no experimental data. They are just the protocols. It means that the people can try to search for some specific protocol which will realize under specific conditions, but they can't access any experimental data. The central storage can aggregate the results from different local storages. So if you imagine that there is one server in one laboratory, second server in the second laboratory, then the laboratories can cooperate through the central storage. This is the way how we can support the cooperation between different local storages. Definitely, the users need some access points to the servers because they don't want to care about what is the architecture of the server and so on. So we have three main access points. The first one we call protocol manager, and this is the software you probably installed on your local computer. What you can do with this piece of software is to design, fill your protocols. You can also do the data processing and some other things. Web interface. This is the platform-independent solution to access the local storage. And we can do the usual data management and protocol management, and we can also do the visualization through the web interface. And the main part is probably the sharing of the experimental work. As the last access point, we support mobile devices, which means that you can access your local storage using your mobile phone or tablet. The reason why we developed this mobile access is that we will try to support the field experiments. So from the aquaculture point of view, you can imagine that you have your phone with you when you are going to the aquaculture tanks. You need to have the computer, and you can check what experiment is running, particularly in the particular tank. So this is the reason why we have mobile support. On this scheme, I would like to show you how is the system connected. So what you can see here is the local repository. This is the local server, which can be placed in your institute. Um, and each user has his own user account on this server, and all protocols together with experimental data are stored on this local repository. Then we have the access points to the local server, and the first one is the protocol manager. Um, so this software is installed on the computer, and what you can do with the software is that you can create your protocols, you can design your protocol templates, you can fill the protocols with the experimental data and the description of experiment. And the software allows you also to communicate with the measurement devices if the device support this possibility. Later on, we will show you how it can be done in a very simple way. The software also supports the standardizations, which means that during the design of your protocol, you can use the standardized terminology to define the individual fields in your protocols. So then you can be sure that you are using standardized terminology in a very simple way. The second thing you can do using the software is that you can run data processing directly in the system. So you can process your data with some specific tools, or you can do the visualization of the experimental data or data from the protocols. So on Friday, you will see some examples of how you can process your data and what kind of visualizations you can do. The last part of the system, which is marked which is highlighted by the red color, is the central part of the system where you can see that it aggregates the data from the local repositories. Not the experimental data itself, but just the description of the experiment. The reason is that this central part is available to any registered user. So the server is not sitting in your institute. It means that you don't have the control over the system, and therefore the user can decide to share just the description of the experiment. So if someone is interested in any kind of experiment, he can try to log into this central part, try to search for the specific experiment, and if this experiment was realized, he can try to contact the data provider and then agree on the conditions which are for the specific data sets. Because I was speaking about the reproducibility, then I should clarify how the system can support the reproducibility. And the way is that we support the terminology that the people are using for the design of the experimental protocol. We also have some standardized way of the description of the protocol, as you will see today. And we try to support standardized procedures. This feature will be developed within this Aquaexcel 2020 project. The advantage of the system is that the data and the metadata, which means the description of the experiments, are stored on one place and they are linked together. It means that you have the complete information about the experiment. We also provide the possibility of the backtracking of the data processing and analysis steps. This means that you have the full control over what was done with your experimental data, and you can simply follow every single processing step. I will show you in web interface how it can looks like. I was speaking about the experimental protocol, and this is the main part of the system. The system is based on the electronic protocol, which means that the protocol contains the information about the description of the experiments. These are the metadata, and also the experimental data itself. So metadata, what we understand as metadata, we understand that it should be the description of the conditions. Under these conditions, you realized your experiment, or it needn't to be the measurement of the data, but it can be also the data processing. So then in the protocol, in metadata, you can describe what software you use for data processing, what was the setting of the software, and so on. Experimental data, I have to mention that it's a black box. What does it mean? Because the system is designed to store any kind of experimental data, because there can be a lot of different data types. Therefore, the data are just the black box for the system. It means that the system don't see inside the structure of the data. The disadvantage of this approach that there is no simple way how to do the queries over the experimental data itself. But the advantage is that you can store any data type to the repository. It means that if your experiment produce the time series, you can store to the system. You can store the images, you can store the videos, you can still store Excel tables, and so on. So the system is not limited in the way of storing of the data types. But the disadvantage is that you can't do the queries. There is the way how the system can see into your data, and it will be explained on Friday. And the way is that you use some specialized data processing module for your data. So you have to know what is the structure of the data, and then you can use the module to see inside the data. So here's the description of how we represent the data in the BioWES system. So first of all, we start with the protocol template. You can imagine the protocol template as an empty protocol where you just described what is critical from the point of view of reproducibility of the experiment. So what are the conditions that you will realize the experiment under? You can imagine that also as the empty Word document where you just described your experiment, but you didn't fill in the particular conditions you are measuring the experiment under. So it defines what should be described for the particular experiment, but it's empty. Then we have the second stage where we use the protocol template for realization of particular experiment. Then we call it protocol because we fill the empty protocol template with the particular values of the experimental conditions. The last part of the data representation are the data itself. It means that we have to somehow upload our experimental data to the system, and the data are stored as the data files in the structure of the system. The user needn't to care about how the data are stored because they are stored in the database. We describe it as binary structure, and the most important thing is that the people can upload any data type to the system. So it can be the experimental data, it can be also the report, it can be PDF files, images, papers, and so on. So the system is not limited in the way of data types. On this slide, I try to summarize the workflow. It means how you can work with the system. So first of all, the user has to install the protocol manager to some local computer, and then the user can start to use the system. The user has to create the template, which is the protocol template, I will explain, for the particle experiment. So at this point, the user should know what kind of experiment he's going to realize. He should know what are the conditions critical from the point of view of reproducibility of experiment, and everything should be described in the protocol template. So for the protocol template, we can use the tool we have, which is called Designer, and I will show you how we can work with the tool. Uh, During the protocol template design, you can use the standardization. You will see how it works on Friday. And the created template is stored to the local server, which means that once you create the protocol template, then you can start to share with someone else, or you can use the template for the realization of the experiment. This is the next point. So when we start to realize the particle experiment, we just simply open the protocol template, which is already created by us, or someone else created for us, and we fill the information about the experiment into the protocol using the protocol manager. Once we describe the experiment, then we also attach experimental data to the protocol, and everything is stored to the local server again. So at this point, we are sure that we have the complete description of the experiment. We have the description of the experiment itself, so what were the conditions, and we have experimental data. Once we save everything to the local server, then we can use web interface to manage our protocols. What can we do with the protocols and with the data? We can download the data from the server to our local computer and do some processing or analysis. We can share the data and metadata based on the access levels we decide for. We can insert the information about the processing steps. It means that we can link the protocols, and we can do the search of the metadata. It means that you can ask the system, show me all protocols where I use some specific fish species, or I work with some specific temperature, and so on. So right now we know what are the parts of the system and what is the usual workflow. So I'll try to introduce you into the individual parts of the system. So the first part is protocol manager. As I told you, that's the piece of software which is installed on your computer, and it allows you to create protocol template using graphical designer. It allows you to reuse the template. So if you have some template for specific experiment and you decide that I would like to do some similar experiment, the software allows you to reuse the parts of the protocol, so you need to create everything from the scratch, but you can use what you already created. Then you can use the protocol template for particle experiment. You can link your protocol with experimental data, and of course, additionally, you can add any other files like reports or papers, so you can have everything related to your experiment to work in the system. And everything is stored to the local repository, because protocol manager is one of the access points to the local repository. The software also allow you to work with the plugins, which is very specialized piece of code, piece of software, which can help you to communicate with measurement devices. It means that if you have some device, for example, LCMS machine, which can export the setting you use for the measurement, then our system can very simply read this information, and you needn't retype it to the experimental protocol, but the system can read and fill the protocol automatically. But it depends on if your measurement device supports some communication. Electronic protocol. I will describe that the electronic protocol is the basic part of the system. How we can create the protocol template? We can use the graphical method for protocol design, where we have predefined components, and we can very simply drag and drop the components to our protocol and create the protocol in this intuitive way. Today I will show you how we can use this designer to create our first protocol template. The output of this step is the protocol template, and it's saved to the local database. The advantages of the approach is that the description of the protocol is unified, so then the system can very simply read everything from the protocol automatically. We can use the standardizations, and we can also use the information from measurement devices. Terminology. Terminology is one of the parts of the standardization. So on Friday, you will see how we can use the particular terminology which was developed under the Aquaexcel project. And therefore, the user needn't to learn the terminology, but he can simply or she can simply start to use the terminology. So this is the main advantage of the system, that you can simply say, download the terminology from internet, and then the system start to offer you the terms during the design of the experimental protocol. This slide tries to explain how the system can provide the communication with the measurement devices. So one of the examples from our laboratory is that we have the microscopes, which can export the setting we use for the measurement. So you can imagine that the microscope has the computer which is connected to, and the computer can provide the simple text file where you can read the information about the magnification of the microscope, about the temperature that was used for cultivation of the cells, what was the camera setting you use for the taking of the pictures, and so on. So through the data file, where all the information is stored, then we have the specialized plugins, which is the software implemented specifically for the particle measurement device. It's very simple software, and it can communicate with the system. So therefore, we needn't to retype the information about the setting of the microscope to the protocol, but we can very simply just click on one button, and the tool will ask for the file where we can see the information about the setting of the device, and then it simply fill the information to the protocol. So the advantage is that it's faster, and it minimizes the errors that can happen during the retyping of the information. Data processing modules. These are the specific piece of software which can be used for processing of your data. I already explained that the system works with the data as a black box. So it means that the system don't really know what is the structure of the data. Um, uh, Therefore, we developed these data processing modules, which are designed for specific data type. For example, if your data are stored in Excel sheet with some specific columns and rows, then we can develop the module which can read the data from this Excel, and the module can do some processing, some statistical processing, or any kind of processing you need. uh, the modules can also provide you the visualization of the data, which is useful if you have some data in the system and you would like to very simply and fastly see the results from different protocols. So you can just click on the module and it read the data from the local repository and show you the graphs. So I will also show you some examples of the data visualization on Friday. Data processing chain. Data processing chain you can imagine as the separate protocols which are linked together to describe the processing steps. It means that usually we start with some data measurement or with the description of some project. Then we do some data measurements, and then we can do different steps of data analysis and data processing. So the system allows you to connect the protocols together, and later on you can visualize this data processing chain. It means that you have the overview of the complete process, and you can simply click on each protocol and you will get the data, and you will get also the description of the experiment. You will see how it works. Web interface, that's the second access point to the local repository. And the main functionality of web interface is that you have the access to the secure data storage. The local server or the local repository needn't to be just the server, but the data can be also stored in cloud or any other storage. So what can you do with your protocols using web interface? You can do the sharing under different access levels. So you can decide if you will share just the data, or you will also share the metadata, I mean description of experiments. And you can decide what is the access level. So the user can just read, or you can modify the data or metadata. You can also use web interface to do the queries. So you can do the searches over the protocols. And you can share your description of experiments to the central system. The advanced features of the system are also the data and metadata visualization, and you can also follow the development of your protocols during the time. It means that if you have one protocol and you change it, you remove some parts, you add some other parts to fit some other experiment, then you can see what was the change in the protocol and who did it. I already spoke about the evolution tracking. Here you can see one of the examples how the visualization of the experimental work can look like. So if you imagine that we have several steps of our work, first we describe the project, then we measure some data, then we do several steps of data analysis and processing, and finally we publish some paper. Then what you can see on this graph is that every single node represents the step in your experimental work. So here we can describe the project. Here we did some microscopy. Here we did some segmentation of the cells, and then we decide about some biocompatibility of material or something else. So this is the way how you can simply have the overview of all the steps of your work you did in the system. Evolution of the protocols, already described that. So if you have some protocol and you modify this protocol to fit some other experiment, then the system remember the information about that the protocol was changed and when it was changed. So then you can see the changes over time. The last access point to the system is the web interface, which is it's a mobile interface, which is the web interface for mobile devices like mobile phones or tablets. And the main purpose is to be able to realize the field experiments. It means that the web interface is optimized to the small screens. And what you can do using this web interface is that you can fill your protocol. You needn't to create the protocol because it's not very handy. You can attach experimental data, usually the pictures or GPS coordinates, and you can send everything to the local server. So it means that from the field experiments, you can have immediately the data in your system, and someone else can start to work with the data. So this was the last slide to introduce you to the system. And of course, if you would like to know or found anything about the system, then you can go to this web page, and there is the full description of the system. You can also download some examples of data processing and so on. The second possibility how to get more information about the system is to go to the Blackboard. Every participant of this training course should get the link and login credentials to a Blackboard system, where you can find all the documents. It means the description of Aquaexcel repository, the help to web interface and also to the software, to the protocol manager, and installation guide how to install the system. So everything should be on one place. So this is just the team who developed the system, and right now we will switch to the real software. So this was the first part where I tried to introduce you into the system, just to understand how you can use the system and what are the main parts. Okay, I'll try to close the presentation. And I will start with the demonstration of the real system. So because it seems that probably all of you were able to register to the system, then you know how to access the web interface. So if someone would like to access the system through the web interface, then you can just write this IP address. This is the IP address of the Aquaexcel repository server to your browser, and you will get this page. So what the user has to do as the first thing is that you have to do the registration. So once you click on the registration, then you have to fill the mandatory information. You have to provide your email. You have to provide your password, and then click on Create Account. At this moment, the email is sent to the administrator of the system, and the administrator will allow or reject the request for the creation of the new user. So once you have the your login credentials, you can log into the system. So you can simply use your username, which is the email, and your password to sign into the system. So this is the web interface where you can do the management of your protocols and experimental work. I'll just try to explain what are the main parts. So the first list you can see is the list of your protocols. If you log into the system, your list will be empty because you don't have any protocols. You can see that I have several protocols already created. And later on, I will show you some examples of these protocols. I explained that the system is based on protocol templates. It means that here is the list of the empty protocols I created for some specific experiments. So you can see also the list of these templates. Once you click on some specific protocol, then you will see what is the content of the protocol. So I will use this biocompatibility protocol, and you will see that we have the basic information about the protocol, which is the name, who is the author of the protocol. You can go to the template which was used for definition of the protocol, and we have some other features of the system you can see. At this part of the screen, you can see the information that is filled in the protocol. So this is a very simple way how you can get the information about what is the content of the protocol. So this is the description of your experiment. You can also click on the data files, and the data files will list you all the experimental data that are connected to the specific experiment. In this case, we have just one file, which is a description of some experimental work. Later on, I will show you that you can attach any data types. What you can do with the web interface is that you can manage your account. So if you click on your name, then you can change your password, or you can see what is the free space on Aquaexcel repository. So you can see that right now we have enough capacity to store your experimental data. Here at the bottom part is the list of the notifications. So here you will see the cooperation with the people who has the account in the repository. So if someone share you some data, it will appear here, and you will also get the email from the person. So you will know that someone is sharing some data with you. The administration part, this is just for the administrators, so they can manage the user accounts, so you will not have the access to this part. And then we can do the searches. So later on I will show you how it works. So this is the web interface, but we have to start with the design of the experiment. So we would like to use the application we installed on our computer. Therefore, we have the software called Protocol Manager. And if you were successful to install the software, then you should have the BioWES icon on your desktop. So right now, I will start the software, and I will show you what are the main parts of the software. So again, I have to log in using my credentials. I have to use my password. It's the same login name and password as for the web interface. If you don't want to log in every time you run the software, you can check login automatically, and next time I'll need to provide the login information. So right now the software logs in to the system, and what can I see? I have two main windows in the software. One is called protocol templates, which means that it contains the list of all the protocol templates I already created or someone is sharing with me. So you can see that here at these protocol templates, I am the owner, and some protocol is shared to me. Protocol template is shared to me. If I click on protocols, then I have the list of all existing protocols. So again, for you, you will have empty list because you didn't create any protocol template and also any protocol. Then for the protocols and for protocol templates, we have the icons here in the top menu. And through the course, you will see how you can use these icons to manipulate with your protocols and protocol templates. So because the first step, if you decide to use the system, is create the protocol template, then I will start with the definition of protocol template. So if you go to protocol template tab and you click on add, then you can create new protocol template. So I define the name test Aquaexcel, and once I click on OK, the new protocol template was created. So what you can see here is the protocol template designer, which is the graphical tool for designing of your electronic protocols. We have two main parts of the designer. Here we have our own protocol with different parts. The final part is dedicated to the attachment of experimental data. So the user can change this part, but later on you will be able to use this part to attach your experimental data. At this part, we have predefined components how to design our protocol template. So you can see that we have different controls like the buttons or numbers or text, tables, and so on. And in a very simple way, we can design the protocol using the graphical designer. The experimental protocol template is good to divide to some logical parts. So therefore, we have different tabs in the protocol template. Each tab can represent one part of the protocol. For example, if you work with fish sampling, then you would like to probably describe the fish species and the fish we use for the sampling, and also we can describe the method itself, so it can be separated to two parts, but it's still in one protocol. So how to create the protocol? If the user needs some specific control, for example, the buttons, then you simply drag and drop, and you place it to the protocol. So this is the way how you can create your experimental protocol. Each specific control has the possibility to move around the protocol. You can resize the control, and you can define the features or the properties of every single control. So at this part of the software, you can see what are the properties of the specific control. Each control has the field mandatory, which means that the user can decide
if this control has to be filled by the user who will fill the protocol later on. So if it's set on true, then later on you will have to fill this part of protocol. If we decide to to not that this part of the protocol is not mandatory to fill, then you define false. Once you change each property of the control, for example, I will change the name, fish species, then you have to press enter, and then the change is applied to the control. So in the case of the buttons, I can, because right now we have just 2 buttons, I can add or remove the buttons to describe my protocol. So this is the way how we can define the protocol template, which means that here we describe what will be the experimental work, but we don't provide any information about the particle realization of the experiment. Once we decide that I have the complete protocol, then I have to save my work to the database. So I click on the Save button, and everything was saved to the local repository. So you can see that the template was saved. Right now, after I save, I can close the protocol. And if I would like to go back to the protocol because it was not finished, then I can still open the protocol. And I can change the protocol. So I can redefine something, I can add some other parts, and so on. I can save the work again. And at this point, I can decide that, okay, I described my experiment well, and it's described completely. At this point, I have to finalize the protocol. The finalization means that the protocol template is locked and no one can change the protocol template. Once I click on the button, then you could see that it was saved, and I can move the controls, I can add anything, I can change the protocol template. If I go back to the list of the protocol templates, then you will see that it was marked by the green mark. It means that this protocol template is finished and it can be used for the realization of the experiment. So right now I have the empty protocol, and if I decide, I can share my empty protocol with someone else. So I can go to the web interface. So I go to the web browser. And if I go to my protocol templates, then I should have my created protocol in the list. So I click on the protocol template. And then the information about the protocol template appears. If I navigate to the sharing options and I click on it, then I see the complete list of the users that who has the account in the repository. And at this point, I can decide to share the protocol with someone else. So I can simply click on the access level, and I can decide what is the access level to share my protocol template. You can see that we have predefined several different levels of the sharing. And for each level, it's visualized what the user to whom I share can do with the protocol template. So here for guest 3, you can see that the user can read basic information, can read metadata, and also can read data files, but the user can't modify anything. For the coauthor, our co-author has the full control over the experiment, so he can read and write everything. So this is the way how can I share something to the rest of the people. So if I decide that guest 3 is the access level I want, then you could see that the setting was saved, and the email will be delivered to Antonin Barta that someone, it means me, share the protocol with him. So this is the way how you as the supervisors can create the protocol template, share it with your students, and the students can just use the protocol template to realize the experiment. So at this point, we decide that it's time to realize the experiment. And it means that we go to protocols and say add new protocol. Again, we have to define the name. It's test Aquaexcel again. And because the protocol has to be based on a specific protocol template, then we have to choose the correct protocol. And we can find our already created protocol template in the list. And once you click on that, it means that the protocol will be created based on this protocol template. Once we click on OK, then the protocol will be created. What can you see here is the empty protocol which we created. We have 3 main tabs. We have some controls in that. And what is highlighted by red color, it means that it's mandatory. So the user has to fill these controls. So right now I start to realize my experiment, and I simply write down into the protocol the experimental conditions I use for realization of the experiment. For example, I can fill in the cultivation conditions, the temperature I use for the fish. I can describe the diet. I can describe the concentration of oxygen, and so on and so on. So this is the way how I can describe my experimental work. Once I describe the experiment, then I should attach experimental data. And as I explained, you can use the last tab of the protocol called data files. Here we have different possibilities what we can do with the data files. And because I would like to add some experimental data files, I click on Add Files. And to demonstrate you that we can upload any data type, I will simply upload some specific data. Then I will upload Excel sheet, some image, some text file, and also some image. Once I click on Open, then the system read all the data files, and it store to the specific protocol. So right now we have the complete information about the experiment. We have the description of the experiment, which is described in the protocol, and we have the experimental data, which are attached to the protocol. So right now I can click on Save, which means that all the data and the protocol is stored to the local repository. So right now I should have everything in my repository. Again, we can check that the data and the protocol exist. Then I can use a interface again. I go to a list of protocols, and I have to refresh because I had the old list of the protocols. And you can see that in the list of the protocols, the test Aquaexcel was created. I double click on the protocol name, and what can I see is overview of the description of the experiment. I didn't fill anything here. And I can see the data files that are attached to the experiment, to the protocol, sorry. Once I decide that I would like to download all the data to my computer, I can click on download all files, or I can just choose one, for example, this image. I double click on it, and the file is downloaded from the server to my local computer. So this is the way how I can get the data from the server to my computer. So if you are connected to the local repository from any computer, you can simply, under your account, of course, read your data and read the description of your experiment. So these two steps are two main steps what you can do with the system. You can simply define your experimental protocol, as I described, using the graphical designer. You can fill the protocol, and you can attach experimental data to your protocol. And once you save your work, everything is stored under your account to the local repository. It means that you can access the protocols through the web interface, as I showed you, or if you installed the software, then you can open the protocol again, and you will see the full appearance of the protocol. You can see what was filled in the protocol, and you can also download the data. So we can download all the data files or just the selected data files. So this is the way how you can later on work with your protocols. Because I just show you some very simple example, then it will be better to show you some real protocol. So for our case, we prepared the protocol from our laboratory, and the laboratory deals with the testing of new materials. They used the time-lapse microscopy and cell cultivation to test if some material, it doesn't matter what is the material, how the cell grow on the material. So I will try to show you the example. Okay. And this is the description of the work that is done in the laboratory. So they do the testing of some biocompatibility of the material. It just means that they have some samples, they put the cells on, and then using the microscope, they are taking the pictures, and they try to analyze how the cells grow. So if they grow well, it means that the material is probably biocompatible. If the cell dies or they doesn't grow well, it means that it has some different level of biocompatibility, or it is not biocompatible. So what they have to do, first of all, they somehow have to describe the material. So it can be one protocol. So we have to decide what kind of experiment we will do with the material. Then they do the microscopy, where they have to describe what was the setting of the microscope, what was the cell culture they use, what were the cultivation conditions. And at this point, we have the image series from the microscope. It can look like this. So these are the cells, and we try to analyze how the cells behave. In the third step, we try to do some analysis of the cell behavior. It can be done by image processing or just manual observation of the cells. So where here we have to describe what method we used for the analysis of our data. It can be some setting of the software, or we can simply describe how we analyze the cell growth and describe in the protocol. The data here is some Excel file where we summarize everything what we did with the cells. We calculate the area that the cells occupies in the image, and this is the information how we can decide about the biocompatibility level. And this is the last step of the work where we describe what is the biocompatibility level of the particle sample. So we can see that we have several different parts of the work, and it means that each work can be described by a specific protocol. So once I will switch to the system, then you can see that I have several different protocols for this particular work. First of all, I have some protocol about the material. So if I open the protocol, then you can see that we have some description of the sample, when it was delivered, who was the responsible person, and we can have the link in the protocol to the specific protocol which describes the sample. So if I click on the link, then the system show me the protocol with the description of the sample. So I have some physical description of the sample, again when it was delivered, some sample ID and so on. So you can see that we can have the links inside the protocols to some other parts of the experiment. Then we have some the other protocols which describe the microscopy itself. So if I open this protocol, then what can we simply see here is the description of the experimental conditions. So we can see the setting of the microscope, what is the resolution of the images, exposure time, what was the temperature used for cell cultivation, or CO2 concentration used for cell cultivation. We also have the experimental data files, and here you can see that we have several different series of the images that are coming from the microscope. Okay. Because we have different protocols for different parts of the work, then the feature of the system is, as I explained, to show you the overall picture of the experimental work. So if I open the main protocol, it means the first protocol of the work, which describes the material and the test we would like to do. And I click on Genesis, then the system visualize the complete list of the protocols related to the work. So just to understand what is in the graph, here is our protocol where we describe the sample. Then we did different types of microscopy. We prepared different types of samples. So we have the protocol which describe one sample, and we have 3 other samples which are delivered, and we would like to do the timeless microscopy on. From these 3 samples, we use these samples for timeless microscopy. So you can see that we have some microscope called BioStation. We did the microscopy. Also for this sample, we did another kind of microscopy. And finally, we analyzed the images, and we did some evaluation. So we decided about the biocompatibility level from this sample, from this measurement and from this measurement. So what you have here is the complete overview of what you did with the experiment. Once I click on some node, then the protocol appears, and I can see the description of the experiment. So you can see what was the setting, magnification of microscope, cultivation conditions, and so on. And also I can access the data files. So I click on data files, and you can see that I have a list of the images. If I try to open some image, then it should be downloaded from the repository. it's not. Probably there is some problem with the connection. Or you can download all the files and to your computer, and you can start to analyze the images. So in the next stage, in the analysis of the data, we have again the protocol. We have the general information, and we have some information about the statistics we did with the cells, and some final evaluation of the experiment. So these are the main steps for the experiment we can do. So we can see that we can have different protocols which describe the individual parts of our experimental work. And we can link the protocols together. And finally, what we can do is to visualize the connections between the protocols, which should describe our complete process. So very simple way how we can have the full overview of what we did during the experimental work. So these are the main features of the system. And right now I'll just quickly try to show you how we can use the individual components to create the experimental protocol. This is the part where you'll finish the first day of the training course, and you should be able to create your own experimental protocol. So I go back to the system, to the software, which is installed on my computer. And I will create another example of the protocol. So you could see that I click on protocol templates, I click on add, and right now I have to define the name. I can test, I'll use date, and I'll click on OK, and the protocol template designer will appear. Right now I will try to go through each individual control and just explain you how it can be used. So first of all, probably we would like to change the names of the tabs. So once you click on the tab, you can define the name. You can say that you would like to describe fish species. So this is the way how we can describe it. If we need more tabs, we can add some extra. We can of course change the position of the tab. So the standard things what we can do. The first component we can use for the design of the experiment is called group box. Group box is the container which can contain other controls, and this can be used to subdivide the page to more parts. So for example, if you would like to describe some diet, then we can define the name of the group box, and we can put some other controls into the diet group box. So for this demonstration, I will use the buttons, and I will change the name of the buttons to diet type. Press enter, and you can see that the name was changed. If I need more buttons than I have, then I can simply click on plus in the properties definition, and new extra button is added. I can reshape the control using the boxes here around. Of course, I can change the names of the buttons, so I can define diet 1, I can define diet 2, I can define diet 3. Every time I press enter, the content of the control change.
So this is the way how I can use the buttons. Um, for the user, for buttons, you can choose just one button at a time. So it's called radio button. So if the user would like to use just one diet, he will just use one of the possibilities. The same principle is used for the checkboxes. So I'll also place the checkbox. And the difference between the checkboxes and buttons is that at one time I can have checked more checkboxes. So just to show you how it really works, here in menu we have the preview button, and if you click on preview, then the protocol is uh, is shown how it will looks like in later on when you try to fill the protocol. So here you can see that I can choose one button at a time, and for the checkboxes I can choose more. The way how I can use the checkboxes is that if you define the protocol for some students, you can very simply define the individual steps they have to do, and during the realization of the experiment, they will just check the checkboxes that they followed the method you describe, and you can be sure that they didn't miss any step. So right now I close the preview, and I will try to describe the other parts. For numbers, I drag drop the number, and what you can see, what I can define are the decimal places. So I can decide that I will need 3 decimal places. I can define the units. For example, I can define centimeters, and I can say if the control is mandatory to be filled or not. And then I can simply use the numbers. Date time. Date time is usually used for the description of the period, so when I started the experiment, when it was finished, and so on. For date time, we can have the different formats. We can use just the date, or we can use also time, or we can use the combination. So I click on the combination, press enter, and you can see that the format was changed. I can also use different formats of the date, and I can use different formats of time. It depends on what you usually use. Again, it is the field mandatory or not. I already explained the checkboxes. You can see it here. And the other component is the image. So image is useful if you would like to show to the experimentalist that there is some scheme. For example, if you have more tanks, then you can mark the tanks. You can provide the pictures of that, and therefore this control has different features. So we can define the name, of course. We can define where should be the label. So let's say that let's say that it will be at the bottom. And what can be done with the image is that we can define if the image is read only or not. It means that if you specify some image here at the design of the protocol, I will do that. So you can choose the image when you click on select image. And for example, I will use this picture of Atlantic salmon. Then you can resize it. Then you can define if the user later on can change the image or not. So right now, when I change it to true, then it means you can see it here in the link, in the hint, that later on the user can change the image. So in the protocol, when the user starts to use this template, he cannot change the image. If I say false,
Then in the protocol, the user will be able to fill some specific image which can represent the data or anything what was done during the experiment realization. So you can upload the picture of your tanks, you can upload the picture of your fish, and so on. So this is the way how to change the image in your protocol. Drop down, that's a specific feature. If you are working with some predefined species or predefined devices, then you can say, okay, so I will probably work with Atlantic salmon, or I will work with sea bass, and so on. And the user needn't to fill anything. The user can just choose one of the possibilities. Uh, Here, the specific property of this control is that if the control is editable or no. So it means that if you say that the control is not editable, it means that if the user use this template for protocol, I'll click on preview again to show you, then you can change the possibilities. I can just choose one of them. I close that. And if I change it to true, then in the preview, you can see that I can choose one of the possibilities, or I can define my own, so it can be some other. So this is the way how I can use drop down. Label, it's the standard thing. If I need some label which is not interactive component, I can place it where I want, and I can label some part of the protocol. Protocol link. I will show you that we can have the links to some other protocols in the system. So what we can do, we can add the protocol link if we need the link to some other specific protocol. For example, if we describe in the protocol that we use some diet, then the diet can be explained, can be described in some other protocol, and in our protocol we can have just the link to the diet. So what we can do with the control, we can disable QR code, which is not necessary. And we can again say if it's mandatory or not. Later on, in the protocol, the user will be able to click on the protocol, and then you can choose one of the existing protocols. So I will show you this on Wednesday, how it works. QR code, you can see the QR code here. The QR code is useful if you will print your protocol and you have some QR code reader. So then you can put your QR code on the tank, for example, and the information about the protocol is stored inside the QR code. So then you can simply read what you need just from the QR code. Of course, we can have the table. It's very useful. For the table, we can define the number of rows. We can define the number of columns as we need. The standard things, what can we do in Word or in Excel, it can be done here. The user can predefine something to the table, for example, the measurement, and once you define something in the table, then later on it will appears in the protocol itself, and the user can change the other cells of the table. If we need to provide some text, then we have two possibilities. We can have the standard text field, or we can have the rich text field. The difference between these two controls is that in rich text we can modify the text. So if we need some specific text, then we can define the color, we can define the size, or we can define that it should be in bold, underlying, and so on. For the standard text, we can just write the text and the user can't change the appearance of the text. And the last component is the hyperlink. Hyperlink can be used if you need to point out the protocol to anything what is described somewhere, to Wikipedia, to some internet sources, and so on. So then you can very simply change the address and BioWES.org. And in the protocol, I will again click on preview to show you how it looks like in the protocol. You can just click on the link and it will navigate you to the specific web page. So this is the way how you can use the hyperlinks in your protocol. I've showed you all the controls you can use for the definition of your protocols. And right now it depends on the specific protocol you would like to describe. Then you will use the specific controls, and you can divide the protocol template to the parts based on your experimental work. So after I finish my protocol, I save everything to the database, and right now the software sent everything to the local repository, and it should be stored to the repository. So this was the way how. Okay, it was saved right now. This is the way how you can define your own protocol template. And for the next training, which will be on Wednesday, the homework is that you should create your own protocol template in the designer to describe some of your experimental work. It will be great if you will use at least 5 components or 5 controls to define your protocols, because then you can test if it works and how it can be used. So at this point, I will finish this first training course. I'm sorry that I was a bit late because it was scheduled to 11:30. Apologize for that. And for Wednesday, it will be organized in the same way. We will try to show you how you can fill the protocol, how you can attach experimental data, and if you create your own protocol templates, then you will be able to describe your own experiment and attach your own experimental data. So thank you very much for your attention. If you will have any problems with the system, or you don't understand anything, you can contact us via the email, and we will try to help you to solve any issues. So thank you very much, and hopefully we will see on Wednesday. If you have any questions right now, then we will be sitting a couple of minutes here, so you can ask the questions, or you can log out right now and log in again on Wednesday. So thank you very much, and see you on Wednesday.